Hello, I am Matthew Case, and my project was looking at IL-27 alpha expression as it controlled by DNA methylation. So neonatal sepsis is the infection of the blood that arises prior to 90 days of life, and it poses a high clinical burden due to its high mortality rate. And this infection can be potentially treated by IL-27, as its absence has been associated with improved control of bacterial growth. And this is based on evidence that's been found where it inhibits T cell subsets as well as certain functions of myeloid cells, which, for example, are neutrophils and macrophages. DNA methylation can be showed down here in panel A. You can see a methyltransferase enzyme is adding a methyl group to this gene here, and then this occurs at a CBG site, and it will effectively block the progression of RNA polymerase, which will therefore inhibit transcription. And then below that in panel B, you can see the IL-27 alpha gene map. And these thick blue right here are exons of the gene, and then these introns are the thin line between. And below the transcription region, we can see the CPG islands where they are found within and around that gene. And this forms the basis for my project because we are actually able to see that it is regulated by DNA methylation, that gene expression. So we got peripheral blood from adult donors and cord blood from neonatal donors to represent each age group. And that blood was processed into monocytes, which were then plated with the 5 treatment groups we have. So that would be media alone, methionine and choline are 2 methyl group donors, and then 5-azacitidine as a methyltransferase inhibitor, and LPS as the positive control. So after 24-hour incubation with those, they had their RNA isolated, and that RNA was synthesized into cDNA, and then that cDNA was run through qPCR for data analysis. So down here in the results, we can see this is our media alone groups for figure 3, and you can see that in CBMCs, IL-27 expression was much higher than that of PBMCs, which is consistent with previous data that tells us that neonates are primarily the time period of life where IL-27 is expressed. So over here in figure 4, this shows our experimental group data. And you can see methionine and choline as our methyl group donors. And methionine has no significant difference between CBMCs and PBMCs with increased IL-27 expression. But in CBMCs, when exposed to choline, they greatly increase IL-27 expression. And this just shows that potentially these methyl group donors are methylating different genes. And methionine has potentially methylating genes that have no impact on P28, while choline may be methylating genes that indirectly, when they're inhibited, have result in P28 actually increasing its expression like we see here. And then 5-azacitidine, it also showed CBMCs to increase IL-27 expression when exposed to the enzyme. And this is potentially because CBMCs are loosely methylated, which is why they are highly expressed in that neonatal stage of IL-27 is highly expressed in neonatal stage of life. So this loose regulation allows for when the enzyme is inhibiting DNA methyltransferases, the remaining methyl groups there's not a lot of methyl groups that have to spontaneously leave for full expression, as opposed to PBMCs, which are tightly regulated. They have a lot of methylation going on. It might take a while before those methyl groups are able to, spontaneously detach and will allow for any increased expression. Although this donor pool is rather limited by number and diversity, so definitely more experiments, definitely more donors are necessary to confirm these results. I want to thank my PI, Dr. Corey Robinson, for his guidance, and Jordan Vance for assistance, as well as WVU for funding my work.